Okay. Yeah. Hi, everyone. I am Eva Simpson, as you all know. I just wanted to say thank you first and foremost to the BSCB for this award and for inviting me to come and talk to you today about my PhD project. And also a thank you to everyone who's PhD. Otherwise, I would not be here with that. So today I'm going to talk to you a little bit about the chromosome periphery in meiosis specifically and how my PhD project looked into this. So our lab group is particularly interested in chromosome ultrastructure and how chromosome structure is key to successful cell division. So as we all know, during cell division, many things can go wrong. You can get misaligned chromosomes, multipolar cells, lagging chromosomes, all of which can affect cell division and lead to issues such as aneuploidy. Despite many advances in imaging techniques over the years, we felt a little bit limited in our ability to investigate 3D chromosome ultrastructure in meiosis specifically, so we decided to use a technique called 3D CLEM. This combines light microscopy and electron microscopy and allows us to, can you see my mouse? Yeah. To create these 3D models of individual chromosomes that you can see here with nanometer precision. From that, we're then able to extract geometric data. For example, we can look at surface area, volume, relative position, compaction. There's an endless amount of data that you can get from these models. In particular, as I said, the component that we were particularly interested in was the chromosome periphery. So if you saw Belle's amazing talk earlier today, you will know a little bit about that. It's essentially a coat that surrounds the surface of chromosomes. Previous work had shown that Ki67 is the key organizer and recruiter of the chromosome periphery. So in mitosis, if we look at some previous work that was done by my supervisor, you can see in these control cells, the chromosomes have this really nice clean structure that is exactly what you would expect during mitosis. When Ki67 is removed, that chromosome structure is altered and they start to form aggregates. And you can see how this could potentially lead to issues with cellular division. So in mitosis, the chromosome periphery is relatively poorly understood, but obviously there is lots of work happening to improve on this. In meiosis, which is what I am interested in, it's pretty much completely unexplored. So this was a really key area of my PhD project in particular. I'm just going to do a quick run through of 3D CLEM because I wasn't sure if everyone would have used it or heard of it even before. So I wanted to make sure that you all had context for the data that I'm going to be showing today. So this is the general pipeline that we would use. The key thing essentially is that you have some sort of marker so that you can track your cell throughout the entire process. So for ours, we were using gridded dishes, which was where we could seed or place our cells of interest and then follow them through the entire pipeline from the light microscopy through to the electron microscopy at the end. Light microscopy in our case was specifically used to identify the rare events that were different stages of cell division. And then once we had identified those and used the gridded references from the dishes, we could process them for electron microscopy. This involved staining, embedding, dehydrating, lots of different steps, and then preparing them for electron microscopy. I will say I think this is the only protocol that any cell biologist has ever been told to use a hand saw on their samples. Unless anyone knows of any others, please let me know. But once that has all been done, we could do some volume electron microscopy. I had luckily access to both a serial block phase SEM and a focused ion beam SEM for my project. And then with those images, I was able to reconstruct chromosome models for the entire complement and then look at them each individually. So one of the first things that we did was actually compare the two different SEM techniques. So I started out with some mitotic cells just because they were more readily available than the oocytes that I would be working with later. So we decided that I would image half of the cell with the serial block face, half of the cell with the FibSem, and then I could directly compare the two techniques on the same sample. This was the result. I was a little bit overly cautious with the serial block face SEM, which is the orange one here, so I did not get the full halfway through, but I was able to determine which would be the best going forward with the project. So generally, the serial block face SEM for the larger samples was more consistent because it used a diamond knife to slice the surface rather than a focused ion beam. With the ion beam, we found that there was sometimes curtaining when you got to the deeper layers, which we didn't really want to happen while we were using such a precious sample type like oocytes later on. And obviously, they're much bigger as well. So we're expecting that timing could potentially be an issue. So milling for the FibSem takes a bit longer, which meant that we had to compromise slightly on the imaging. So going forward, we decided to continue with the serial block face SEM for such a large sample size. So before I started this project, I had never, to be honest, even seen an electron microscope before. So to get used to the workflow and to continue on with some existing work that had been done in the lab, I started work on what we like to call the mitotic atlas. So this was in collaboration with the University of Edinburgh, Bill Earnshaw's lab. And what I did was imaged multiple cells at each stage of mitosis. We didn't want to use any kind of drug treatment like nicotazole or anything like that to synchronize these cells. We wanted them to be as pure as possible before processing. So there was lots of searching around on dishes for different stages of mitosis and then processing them for electron microscopy. So you can see the kind of images that we were getting here from the volume EM and then the chromosome complements that we could reconstruct after. And as I said, we could look at individual chromosomes within that. This all came together with the group from the University of Edinburgh, and we were able to get a paper published, which was really nice, that sort of summarized all of the ultrastructural information of each of these stages of mitosis and explored the whole complements themselves, but also individual chromosomes. There's so much more to this paper than the tiny little glimpse that I've just given you there. So if you want to go have a look at it, I would recommend it. It's a good read. Obviously, as I said, my interest was specifically in meiosis, but using oocytes presents its own challenges. As many people have said already, it's an incredibly precious sample type. This isn't something you can just culture like a standard cell line. It requires animal models or patient donations. They're also incredibly large cells. So if you look at the mitotic cells that I was using previously, they have a diameter of roughly 20 microns, whereas the meiotic cells have a diameter ranging from 80 to 120 microns. We're also interested in the chromosomes inside these cells, and in mitotic cells, the chromosomes take up roughly 80% of the total volume, whereas the chromosomes in meiotic cells are only taking up about 10% of that volume. So that just was showing how critical it was for us to make sure that we were targeting our electron microscopy in the right place. And this is where 3D Clem was really shining. We were able to use 3D Clem not only to identify the stages of division that we were interested in, but also specifically where in the oocytes they were present so that we could target our high resolution electron microscopy imaging more clearly. So the first question that we wanted to ask is going to sound like a very simple one. Is there a chromosome periphery in meiosis? The reason that we wanted to know this is most of the information that we currently have available to us on the chromosome periphery is based on mitotic cells. So we wanted to confirm whether or not there was even a periphery present. We used Ki67 to do this, and fluorescent imaging was done on the chromosomes, and you can see here quite nicely that Ki67 did localize to the surface of the chromosomes, and it appeared that there was, in fact, a periphery. Something that we weren't expecting, but that was very interesting to see, actually occurred during anaphase. There seemed to be some sort of asymmetry with the Ki67 distribution in that more of the Ki67 was retained within inside the oocyte than was ejected out into the polar body during anaphase. This seemed to indicate that there was some sort of regulatory pathway that was retaining enough Ki67 for future divisions and development. Once we knew that there was a periphery, the next step was, of course, how can we remove it? So morpholinos were used to knock down Ki67, and you can see from the imaging here the loss of that periphery around the chromosomes. This was also confirmed with line scans of the electron microscopy. So in the electron microscopy, you can sometimes see a little fuzzy coat surrounding those chromosomes, which is the chromosome periphery. And with the fluorescence and the electron microscopy analysis, we were able to confirm periphery loss. Once we had removed that Ki67, that's when we started to see phenotypes in the meiotic cells. So here you can see this control cell progressing exactly how you would expect through meiosis. So it goes all the way through, a polar body is formed, and then it actually realigns back into the metaphase plate where it will remain until it's fertilized, exactly what you would expect to see. In comparison, the Ki67 knockdown cells had various different phenotypes occurring. The first one being that some of the cells just stalled. So they were they were progressing through meiosis 1 up into a point, and then they seemed to get stuck, often in metaphase, and just weren't able to progress past that despite the extended imaging time. The second phenotype was these misaligned chromosomes. So they sort of started to align themselves along a metaphase plate, and then as time went on, they just became more and more disorganized. And you can see, I'll show you the static image in a second where you can see it a bit clearer, but you can see them start to just spread out throughout the cytoplasm and lose any kind of organization. The final one was these missegregated cells. So this one actually seems to progress through meiosis 1 to the point where it forms a polar body. The mistake that it makes is that it actually pushes all of the chromosomes out into the polar body and retains none of them inside the cell. So you can see that a bit clearer here. In the control cell, by 630 minutes, it's divided, it's a polar body present, and then by the end of the imaging, they've realigned along that metaphase plate. At the same time point for the knockdowns, none of them had divided, and in fact, the only one that had managed to divide by the end of the imaging had done it completely wrong. So this showed us that the loss of the periphery was not just a subtle defect, it was actually detrimental to the successful division of these oocytes. Once we had seen these phenotypes, we then wanted to explore this in a little bit more detail and the ultrastructure of the chromosomes, and that was where the electron microscopy came into it. So we did volume electron microscopy of the control and the knockdown cells. As you can see here, you could really nicely make out the different chromosomes inside and the zone of exclusion surrounding them. And then from this, I was able to generate the reconstructions of the chromosomes. So in the control, what you can see here is a really nice, clean metaphase plate, exactly what you would want to see. They're all nicely organized. In comparison, on the Ki67, we could see slight disorganization and then a lagging chromosome, which was exactly what we had seen in the light microscopy earlier. The other really useful thing, which I think I've said a few times now, is that actually you can look at individual chromosomes. So you don't just have to look at these as a whole complement. You can break this down and look at each one separately and compare those. So I used this to use volume data and the known DNA content, oh, it's really tiny, sorry, to essentially generate a karyotype of the chromosomes inside these cells. So it's a bit easier with the smaller and the larger chromosomes because there is much more differentiation between them. So I was able to be quite confident with the first 4 or 5 and the last 4 or 5 that they were definitely the chromosomes that I was seeing. It's a little bit harder with the middle ones just because the volume and the DNA content overlap so much. But with this karyotype, it enabled me to look a little bit further into the smaller and the larger chromosomes. When we did that, we found that there appeared to be some sort of issue with the spatial organization. So in a standard control oocyte, what you would expect to see is the small chromosomes localizing close to the center of the metaphase plate. So this green dot is the center of the metaphase plate. Remember as well that this is all in 3D, not just 2D. So yeah, you would expect to see the smaller chromosomes localizing closer to the center of the metaphase plate, whereas these red chromosomes are the larger ones, you would expect to see them further away. when we looked at the knockdown cells, we could see that there was some sort of disorganization with this, that the smaller chromosomes were more spread out and the red ones had started to come, the larger ones, sorry, had started to come closer in. And when we actually analyze this quantitatively and the distances between the chromosomes in the center of this metaphase plate, we could see that in the knockdowns, there was a significant increase in the distance of chromosomes from this alignment, showing that Ki67 in the chromosome periphery seems to have some sort of role in spatial organization of chromosomes and is not just structural. Another interesting thing that we saw was actually the effect that the loss of the periphery had on chromosome interaction with the rest of the cellular components. So this area that you can see here is essentially a zone of exclusion. So you've got your chromosomes inside, and then you would have your meiotic spindle in here, and then an organelle-rich layer surrounding that. In the control cells, it's a bit clearer to see in this model. That seemed to be a really clean, defined area, and there was little to no interaction between the chromosomes themselves and the surrounding organelles. When Ki67 was knocked down and the periphery was lost, those chromosomes started to encroach into that organelle-rich area. So you can see in the model here, it looks as though the organelles are actually starting to encapsulate that. This suggested to us that there was some sort of role of the periphery as a buffer between the chromosomes themselves and the external cellular components. And we're not sure as of yet whether this is a physical or biochemical buffer, but it's definitely something that should be explored going forward. So from this and much more, me and Ben from Newcastle have a paper in preprint at the minute, which confirmed that the loss of the periphery affects chromosome organization, chromosome segregation, and also interactions with the cellular environment. Finally, the next step of this work was to start to look at human oocytes. I will put a caveat to this. Turns out they were a lot harder to work with than I thought, and a lot of issues came up during processing that we were not expecting, but we did actually manage to do some nice imaging. So I started off with the light microscopy. So if you can just about see it there, that is some chromosomes inside the human oocyte. Took that on to electron microscopy, and the yellow arrow there down at the bottom just shows those chromosomes, and the blue arrow shows the polar body that formed. We were really excited by this image because we felt like we could see the chromosomes really nicely. That's really fuzzy on that screen, sorry. You see the chromosomes really nicely, and we could also actually see kinetochores around the edge, which we didn't think was something that had been shown before, so we were really excited to see that, and even started to see some of the microtubules present connecting to those kinetochores down at the bottom. So I did manage to actually image quite a few human oocytes using this technique. And you can see one of them here. And then this is the nested region, which was slightly higher resolution with the chromosomes themselves and the microtubules present inside them. I was then able to model those chromosomes and even some of the microtubules. So I will say there's a caveat to human oocytes that they are donated by patients, but the ones that we are received for research are generally not considered viable for clinical use. So you do have to be aware that there may be some changes with them. And I think these chromosomes were not the happiest because they looked a little bit strange. But you can see in this cross-section here that we were actually able to start to model some of the microtubules that went through the center of those chromosomes and actually model the chromosomes themselves. There is definitely a lot more future work to do with human oocytes, and during the course of my project, I didn't really get to explore any kind of Ki67 work with the human oocytes specifically, but it's definitely in the next steps. So overall, during this project, we managed to confirm that a chromosome periphery is in fact present in meiosis. Ki67 is a key component of the meiotic chromosome periphery, and also that the chromosome periphery is functionally critical because when we removed it, it was either disorganized, completely unable to provide, or just really unhappy. So there's definitely more work to do in this area, but we feel like we started to lay the foundations for further research into this. I'd just like to say thank you to everyone that collaborated on this project. A special thank you to my supervisor, Dan, and also to Suzanne and Ben at the University of Newcastle, as well as Andrew Carris and Alexandra at the University of Warwick. Any questions?